Hello and welcome to today's lesson. Today, we'll be talking about a crucial genetic sequencing method called pyrosequencing. Developed during the early 1990s, pyrosequencing has significantly impacted the field of molecular biology. It's a method that works by adding and incorporating nucleotides, which can measure the natural release of pyrophosphate in real time, converting it into a light signal. Pyrosequencing can determine specific genetic variations such as single nucleotide polymorphisms or SNPs, insertion or deletion mutations, gene copy number variations, and even DNA methylation changes. All of these are essential for understanding genetic disorders, infectious diseases, and cancer, among other applications. Stay tuned and let's dive into the exciting world of pyrosequencing. Before we dive into the exciting mechanism of pyrosequencing, let's take a quick look to revise how DNA is synthesized. During DNA synthesis, when a new DNA strand is built on a template strand, a phosphodiester bond is formed between the last nucleotide of the growing strand and the incoming complementary nucleotide. However, with the formation of the phosphodiester bond, a molecule called pyrophosphate is released. In pyrosequencing, the released pyrophosphate is then used in a series of reactions, starting with the sulfurylase reaction that converts pyrophosphate to ATP. The ATP is then used in a reaction catalyzed by luciferase, producing light as a result. What's interesting about pyrosequencing is that it doesn't require any form of electrophoresis or separation procedure. This means that it's much faster than other sequencing methods that rely on the separation of DNA fragments, such as chain termination sequencing. Stay tuned to learn more about this rapid and exciting technique. Bisulfite converted pyrosequencing is a powerful technique that was developed in 2003. This technique is primarily used to detect biomarkers for DNA methylation, which is essential in diagnosing and predicting cancer outcomes. Using pyrosequencing, we can effectively detect methylation levels of individual CpG sites in a PCR product obtained by primers common to methylated and unmethylated sequences after bisulfite conversion. This technique has been instrumental in genetic testing, as it can help identify and research genetic disorders, spontaneous mutations, and even transgender research. Additionally, it's been utilized in prenatal diagnosis for various genetic disorders. In cancer diagnosis, pyrosequencing has been used to identify cancer mutations, which assists with identifying the stage of the disease and the best course of therapy. Lastly, it has also been used to identify gut, oral, and other microbiota flora in gastrointestinal diseases, including inflammatory bowel disease or IBD, celiac disease, and metabolic disorders. Overall, pyrosequencing is a valuable technique that has revolutionized several fields within molecular biology in recent years. By detecting DNA methylation biomarkers and mutations, this technique has significant potential in diagnosing, predicting, and managing various diseases and disorders. Pyrosequencing is a four-step process that involves annealing, nucleotide incorporation, signal generation, and neutralization. Like the chain termination method, we need to start with single-stranded DNA molecules that are prepared by a chemical process called alkali denaturation of PCR products or recombinant plasmid molecules. Then, a primer is attached, and the template is copied by a DNA polymerase. Importantly, no added dideoxynucleotides are used during this process. During pyrosequencing, every time a complementary nucleotide is incorporated into a new strand being synthesized, a molecule of pyrophosphate is released. Here's how the molecule is detected in pyrosequencing. Pyrophosphate combines with adenosine phosphorsulfate, and with the help of enzymes called ATP sulfurylase and luciferase, it is converted into ATP and light. This means that the pyrophosphate released during DNA synthesis can be detected through the emission of light. To avoid false signals of pyrophosphate, an enzyme called aparase continuously degrades unincorporated nucleotides and ATP. Once the degradation process is complete, another nucleotide is added. The process of adding nucleotides continues sequentially until we get the desired DNA sequence. To keep track of the nucleotides being added, a substitute molecule called deoxyadenosine alpha thiotriphosphate, or DATP alpha S, is used instead of natural deoxyadenosine triphosphate, or DATP. This substitute is efficiently used by the DNA polymerase but not recognized by luciferase, eliminating the risk of false signals. As the process continues, a complementary DNA strand is built up, and the nucleotide sequence is determined from the signal peaks in the pyrogram trace. The detector picks up the intensity of light emitted during the process and uses it to infer the number and type of nucleotides added. For example, if 3 cytosine nucleotides are added sequentially to the same fragment of DNA, the light emitted will be 3 times more intense than fragments of DNA with only 1 cytosine nucleotide added. If no light is emitted upon the addition of cytosine, then the complementary base in the single-strand DNA template must be one of the other 3 nucleotides. Overall, pyrosequencing is a rapid and precise DNA sequencing method that has significant potential in diagnosing and predicting various diseases and disorders. Given its advantage, it's no wonder that biotech companies like Chagen, Biotage, and Lumidos are now offering pyrosequencing services to their clients. In conclusion, pyrosequencing is a powerful and efficient DNA sequencing method that has revolutionized the field of molecular biology. While there are limitations, its advantages make it an attractive option for researchers and clinicians alike. Thanks for watching.